Okay.
I know, sorry about that, guys. All right, so just if we just quickly review some of our steps. So steps assignment and method. People really think elephants on cards. And those letters that are along each step. Here's the problem, R is research, use hypothesis, use experiment, O observation, and C conclusion. And so yesterday what we did is we started talking about, okay, those are the steps. What does that actually mean? So what do we do in each of those steps? So in the problem step, we start with a question. The question that we're asking. So I just want to answer the question. In the research, we find some background information about whatever topic we're going to be studying. And then we're to the hypothesis stage. And again, we're using a new definition probably for hypothesis. Not an educated guess everywhere. And what do we say? What definition are we ready to start? Start to use. So here, prediction. Prediction about what? Prediction about who's going to win the Yankee game tonight? No. Prediction about what's going to happen in the experiment. Okay, so let me set up an experiment. The point of an experiment, the purpose of an experiment is to test something. It is to test what? Not quite to test the problem, it's going to help you answer the problem. To test the hypothesis, that's right. The procedure that we use to test our hypothesis. So we set up this test. It may take 20 minutes, it may take 24 hours, it may take 6 weeks. Depending on how we set up our experiment and what we're actually studying. As this experiment is going along, or in any experiment, then we need to make some observations. And in these steps, the observations are when we collect our data, to gather our data from this experiment. And once we have that data, we record it in a data table. After the data is collected, the experiment part is yours. Then we need to look at the data, and we need to analyze the data, and that's going to help us with the next step, which is our conclusion. What do we do in that conclusion phase? Yeah? In a way, let's see. So you start to get the data as the results of your experiment, and you use that data for the conclusion. What do you do, Colin? Sort of. Basically, if you look back at the hypothesis, in the conclusion stage, you say, well, I made a prediction back in my hypothesis. Now your conclusion is my hypothesis correct? Is this data that I had from my experiment? Does it support my hypothesis? Was I correct? Is it okay if your hypothesis is wrong? Yes. Have you still learned something from your experiment? Yes. So it's okay. We never go back and change our hypothesis to agree with our data. I mean, we believe our hypothesis, we just say, no, in this case, my hypothesis was not supported. And so that's what we do in these various steps. So let's think about an actual example. Let's think about, that's a rat, right? Let's think about an experiment. It's about an experiment in which we are going to ask a question. And the question we're going to ask in this example is, I guess it's mice. Few mice grow larger if they're given extra vitamin C. You can ask that question. That's our problem. I don't know the answer to this question. There you go. Anyone else? All right, so let's say this is a problem, and we're going to try to answer that question in our experiment. What kind of research might I do before I can really get into this and send it out and research? Really?
See what other things.
I need to know what mice need to survive. Okay? Okay, what foods contain vitamin C and how much? I probably want to know that. What? Well, that's something I'm sort of trying to find out in this experiment, right? So we may not know that yet. Anything else you have to know? Maybe you want to see what our people try to experiment. What? We're going to get to that in a minute. So that's going to be our initial research, but we're going to need to know that. Yeah, these are all things. We got to know all mice, about vitamin C, what kind of diet they usually eat, how much vitamin C they get in their normal diet, what they need for living space, how big are the cages going to be, how much water are they going to need, what types of mice are there. These are all sort of little details that are important to think about before you actually set up your experiment. Okay, so I find out all this various information. I know how to keep mice in the classroom, and how much water they need, and all these other things. It's time for me to make a hypothesis. What are the possible hypotheses? Yeah? Okay, that's one possible hypothesis. What's another possible hypothesis? See? Okay. Although, probably that's not the question I'm asking, right? I'm not asking will mice survive if given vitamin C. I'm asking something specific about their growth. So, in my hypothesis, we're not going to touch that. Here's another thing. We often will phrase a hypothesis as an if, then statement. So, when I give you a hypothesis, can you say it a little differently as if, then? So, if then see you get included in that. See? They will not go to that. So we often will phrase it as if I do this to this subject, then I think this will happen. So if I say you're not going to see, then they will grow to a larger mass. So you guys have to grow larger. We have to be a little more specific. What does that mean? A larger mass, will they get longer, or will they get wider? So we have to be specific. In my example, I said that the mass will increase or grow to a larger mass. Okay, so how do I actually do this? How do I now test my hypothesis? Anyone want to say what I actually do? I give me the whole starting from I'm in this classroom. What do I need to do? One mouse. So first I need what? One mouse? Two mouses, two mice, please. Okay? What else? All right, so some buzz, give me some more. Okay. Same amount of vitamin C? What do you think? So I have two, how many cages do I need? I have 2 cages. What's in each cage? A mouse in each cage? Okay. And we'll go, so I've got the water. So you got food. Okay, what else? Here? And a lot of water. Should their cages be the same size? Should they be made of the same material? Yeah. Is everything in the identical in these cages? What's not going to be identical? The amount of vitamin C. So how am I going to do this? So I can't really give them 2 different foods, right? Because that would interfere. That's an extra variable.
One more and then drops in
Yeah, maybe I give them the same amount of dry mouse food, but I put some vitamin C drops on like that. So really they're exactly the same, except one's getting that extra vitamin C. So I have 2 groups. One group gets a normal amount of vitamin C, and one group gets twice as much, or another gets 3 times as much. I can decide on that. So then it's time for me to make my observation. What observations am I going to make for this experiment? What observations are there? Alan?
the size of each mouse sequence from the other.
Okay, let's just get a small amount, a little bit more specific. Not size is kind of general. How can we be specific, Alan? Yes. I'll be measuring the mass of these mice. That's the data I'm going to collect. Maybe after 2 weeks, I find the mass of these mice. And let's say, as an example, the mice that were the extra vitamin C were about the same mass. That's my data I'm collecting. So I need to use that data to come up with my conclusion. So based on what we've said so far, what do you say? What is our conclusion to this experiment? The way we set it up and propose. Okay. Sure. Yeah, in this case, my hypothesis was not correct. Data shows that my hypothesis was incorrect. The mice being extra was eating after life. So this is how we use the steps of the scientific method to actually answer a question. Now, the way you guys told me to set this up, I don't know if I would trust the results of this experiment. I've got to improve this and make it a more valid experiment. Okay. Okay. You got to review it. That's one idea. The other idea sort of goes along with this, yeah. Yeah, one mouse. I said one mouse in each cage. Well, one mouse may be just genetically a little bit bigger than the other. So what would make it better here? Yeah, if I had 10 mice in each cage. different pages of each type of noise. Then am I going to be more sure of my results? Yeah, because even if one happens to be a little larger genetic or whatever, you're studying that. So it's going to average out a little bit. Okay? What would be even better than having 10 in each case? Okay, could be. I'm not sure about that. But what's better than 10? 20. What's better than 20 miles? 100 miles. Okay? We call that a sample size in an experiment. The greater your sample size, the more confident you can be in the results of the experiment. Okay, so we always want to try to have a large sample size. Okay. All right. You guys ever use the scientific method? Yeah? You go through the steps and say, okay, my problem is this. My research, my hypothesis, here's my data. Probably not. But you do use it all the time. This is one example. How do you use the scientific method? What's the relationship to? All right. What? Okay. Do you know the hypothesis? Okay. Yeah. Maybe if I jump across, in this way, maybe that's going to make me successful. Maybe if I call you, I camp out in this location, I'm going to be able to do my match if I do that. And what do? You try it out. What kind of data do you collect? Are you being successful? Did you come up with a conclusion? So that didn't really work. Let me just come up with a new hypothesis. So we try something else. If you're golfing, maybe do the service, or your grip. Maybe adjust your grip one way. So is this going to help you? And maybe you adjust it. Do an experiment. You go to the driving range and you hit some balls. Collect some data. Say, okay, how is this working? And you come up with a conclusion. Then what is it doing after? Trying to bend the soccer ball to the outside. Maybe another time I try different things. Like try to strike the ball in different areas and experiment, collect some data, and make conclusions. So we use this all the time. There's no way to think about it, but really you are using the scientific method. Now, when we use the scientific method, when we set up an experiment, you guys told me yesterday we have variables, these different factors. Well, we have different categories of variables. There are 3 categories of variables that we talked about. Independent variable, dependent variable, and variables held constant. Has anyone learned about these 3 types of variables before? Okay. Did you learn about variables at all? What do you call the variable in the experiment you change? Did you call it a manipulated variable? You're not calling it. Alright, so let's just go. Alright, in an experiment, an independent variable. Okay, so first variables are factors, things about your experiment that you could change or alter. And the independent variable is the thing you're testing. It's the thing you're changing. Then you have a question about it. And in a properly designed experiment, how many of these independent variables should there be? 1. So if you think about it, the I in independent looks like one number. 1. Because it's the one thing in the experiment that you change, that you're testing. That one thing that's going to be different. You think about it as the cause. You think about an experiment as something working on cause and effect. The independent variable is the cause. So if you think back to the lice example we just talked about, was the independent variable in that experiment? The amount of vitamin C there. That's the one thing I changed. The one thing that was different in my 2 pages of life was how much vitamin C there was. What about that experiment yesterday? We talked about. In that experiment, what was the independent variable there? In the color of the light. When we fixed the experiment, that was the one thing that was different. Okay? Well, what do you think about this? What are variables of constant? I didn't ask more about variables of constant. Okay? Yeah, they're almost little things that we try to keep the same. Things that we don't allow to change. What was one of them in the mouse experiment today? What's one variable we would have held constant? Yeah. The amount of water we gave them. What's another variable we would hold constant? Well, size of the cage. But what other? The age of the mouse. We can think of probably dozens. The species of mouse, and male or female mice, and the temperature of movement, all those things. Those are all variables on the concept. Okay, that leaves us with one other category, the dependent variable. This is easy to remember because D, dependent variable, this is the data you're collecting. In your experiment, the data you collect, the results of your experiment are the dependent variable. Remember the D for data, the I looks like a 1 because it's the one thing you change. I think helps you remember the variable. Again, it's what you don't know it ahead of time. I did know how much vitamin C I was getting in each mouse, but I didn't know the mass they were going to be at the end. So you don't know the dependent variable ahead of time. Something you find out as your experiment's going along. Think of it as the results of the experiment. Okay, last slide today. Now, in my mouse experiment, why didn't I just make it simple? I should have one cage of mice, and I gave them extra vitamin C so how to get it out. Why didn't I just do that? It's simple. Right? Yeah. I need 2 groups so that I am able to compare them. Right? I just had one group, and I found their mass. I said, well, all right. Is that bigger than they normally would be? I don't really know. I need to always in the experiment have at least 2 groups. We call one of those groups the experimental group, or sometimes more than one, and the other group is called the control group. The experimental group is the group when you change the independent variable. You alter it. You gave them more vitamin C, or you change the color of the light. And then you have the control group. The control group is the group you use as a comparison, kind of like a baseline. You could think about it as the control group, you just sort of treat it normally. you don't alter the independent variable. So if you think of the mice, what was my control group? My mouse. Jason? Yeah, the mice didn't just the normal food. And so then the experimental group, what? Yeah. Can I have more than one experimental group? How can I set up my mouse experiment and have multiple experimental groups? I can get some more information. Yeah?
Okay. So here's the experimental, like one group, they can have a certain amount of vitamin C and another can have more.
Exactly. I could have my normal amount of vitamin C, then I get twice as much, 4 times as much, 10 times as much. Each of those are experimental. And then I might know a little bit more at the end of that experiment versus just regular and extra. All right, let's do one example. Actually, I know it's goes last today. Let's talk about this example. So, the JV football team. Let's say the mothers of the Hartford JV football team decided they had it with trying to wash uniforms, wash the stains out of their son's uniforms. Okay? So they decided they're going to conduct an investigation of stain removal. Do you know what stain removal is? It's not like the powder you put in the washing machine with like the extra stuff you spray on or put on with stick. So they washed 30 filthy uniforms with spray and wash. They washed 30 uniforms with game. They washed another without any stain removal. Each load of laundry was washed in the same washing machine, same cycle, 120 degrees Fahrenheit water with equal amounts of soap. And then the mothers looked at the uniforms to see how clean they were. So in this experiment, what's the problem? What's the problem here? Okay, well let's phrase it as a question, because we want our problem to be a question. The question we're trying to answer. Yeah, and a little more specifically? Yeah, we're going to say which stain remover works the best. Okay, in their experiment, what was the independent variable? Alan? No, because they were all equally dirty. Remember, independent variable I is like a 1, so one thing different in your group. So what's different, Gage? The type of stain removal is different. What was the dependent variable in this experiment? Dependent, the D stands for data. What data were they collecting as this experiment was going along? Yeah? What did they look at? What data did they collect? You're not on the right track. I might just phrase it a little differently. What did they look at? Because you sort of made me question how well they were. How do they know which one were better? They look at, how clean. Or two variables they held constant. Two variables they held constant. See, how much one? What's that? Yeah, how long they're in the washing machine? What's another one? Here? The water temperature. Yeah. All right, this is your homework I'm giving you. It's from back. You probably want to take the notes home to do it so that you remember variables and groups and you don't already know that. I will check it in, everybody in class, make sure it comes to class with you, don't leave it in a locker, don't leave it at home. Have a good day.